Welcome, everyone, and thank you for joining us today on International Epilepsy Day. We're pleased to have as our speaker today Dr. Louis Dang, who's the Director of Research for the Division of Pediatric Neurology and a pediatric epileptologist at CS Mott Children's Hospital. Dr. Dang and his team of researchers study the cellular and molecular mechanisms behind genetic epilepsies, with the goal of developing new therapies. And we're going to learn more about his research and other promising research in epilepsy today. So Dr. Dang, I'll hand it over to you.
Great. Thanks so much, Russ, for having me. And thank you to all of you for listening. And if you have any questions, please just write them down or write them in the chat, and I'm happy to take some questions at the end. So the title of the talk is Basic Research in Epilepsy, Unlocking the Future. I'll be talking mostly about basic research, but a little bit about clinical research as well. So just to start really broadly with what is research? What do we do in research? What's the goal of research? So research is defined as a systematic investigation, including development, testing, and evaluation designed to develop or contribute to generalizable knowledge. So basically it's studies that we can use to generate knowledge. Have new knowledge for the world, and that new knowledge should be generalizable in some way, meaning you might study a population of people, which you can generalize to the greater population of people, or you do some studies in mice that you can hopefully generalize to all animals or to humans or something like that. And there are lots of different types of research, but the three main types are descriptive, comparative, and experimental. So descriptive is just kind of self-explanatory, describing the situation of something. For example, that might be, how many people in the world have epilepsy? That's a descriptive study. You're describing how many people have epilepsy. Comparative might be that you're comparing two different groups. So that might be, for example, what is the rate of sleep problems in people with epilepsy compared to people without epilepsy? You're comparing two different groups, but you're not doing anything to interfere or intervene with those groups. And then lastly, it's experimental. So that typically implies there's some sort of intervention or change between two groups. That might mean, for example, I have a group of mice that have a genetic abnormality and they have seizures, and I'm treating one group with drug A and a second group with drug B and a third group with nothing or just some sort of control. So I'm doing some sort of intervention on those different groups and then comparing them after that intervention. And we should just remember that every research study should go by the scientific method. And this is kind of funny to say, but medicine and research wasn't always using the scientific method. This is actually something that had to be invented, centuries ago, but had to be made. And I think the most important thing is, what is your question? So you have to ask your question, and that research has to be focused on answering that question. Once you have a question, a research question, then you do some background research, and then you make a guess or a hypothesis. You make an educated guess on what the answer to your question is based on what your background research has been so far. And then you can test that hypothesis, that guess, by doing experiments, and then you analyze your results. From your results, you might be able to draw a conclusion, and then you report results back. And of course, sometimes you can't draw a conclusion, and then you go back to one of the previous steps, and you might have to do more background research. You might have to reconstruct your hypothesis or do more tests with different experiments. So that's a scientific method on how to basically discover new things. So what is basic research? And with that, what is clinical research? And is there anything in between? So basic science, when we refer to basic science, we typically mean fundamental science, understanding the fundamental things in biology, physics, chemistry that make up our world. And some people also refer it to as bench science, because a lot of the science happens at a lab bench. There's a lot of computational science that is very basic, meaning based on math and computers and modeling, you might be able to study and generate new knowledge. And basic science often utilizes models. Some of those models might be animal models, for example, where we try to get a sense of a disease process or of a biological process, for example, not studying it in the population that we're actually interested in, for example, in humans, but studying in some other animal that we can manipulate experimentally, like mice, rats, rabbits, zebrafish, flies, things like that. Basic biomedical science is typically basic science that is more disease-oriented, which is mainly what we do. There's a branch also called translational research, and that's basically taking basic science discoveries and trying to translate them, meaning move them over to the clinical side, into human subjects, and hopefully eventually into treatment of human patients. And then there's clinical research, and there are two main types of clinical research. So this is human subjects research, and those can be observational studies where you just kind of see what happens and you don't actually do an intervention, but you can still do comparative research and compare different groups. And then there are clinical trials or interventional trials where you're actually doing something, whether that be treatment with a medication, treatment with psychotherapy, treatment with a medical device, or something like that. So those are the different types of research along the basic to clinical frames. And some other examples for basic science, the question might be, in mice, how do neurons signal to each other? It's just a fundamental thing on how biology works. You could also say, is neuronal signaling abnormal in mouse models of epilepsy? So it's also a basic science question, but it's more disease-oriented, so it might be more basic biomedical science. And then you can then see whether specific pathways be targeted to alleviate seizures, to improve the seizures in those mouse models. Translational research then might be, can we apply the studies from these animal models for use in human subjects? And then clinical research might be, is disrupted sleep in children with a specific epilepsy syndrome, Lennox-Gastaut syndrome, is that disrupted sleep associated with a higher seizure burden? So that might be an observational study versus an interventional trial might be, what is the effect of drug X on seizures in children with Lennox-Gastaut syndrome? I think it's also useful to think about research, and especially research as related to therapies in a broad spectrum and timeline too. So basic translational clinical research are all needed for therapeutic discovery. And basic research depends on lab models. I mentioned animals. I'll also talk about stem cell models or induced pluripotent stem cells. Often with that model, we then have to develop some sort of an assay. Assay is a fancy word just to mean some sort of a test or measurement. For example, for animals and seizure studies, we might have EEGs on the animals, and the assay might be, is the animal having seizures or not? So that might be one of the electrophysiological assays. But there might also be other markers or blood levels like biomarkers or chemical assays where you do measurements with chemistry to measure different chemical levels, and that might be what you use to show success or failure of different treatments. Once you have a good assay, then you can screen a bunch of drugs and you can see which drugs fail or succeed in those assays. And then that can lead to preclinical studies where you use those lead compounds in different models. And that's all basic science research. Then you get into translational research where you take these studies and apply them to patients. Typically even before applying it to patients, we need to know what the natural course of disease is. So those are natural history studies, and that really helps guide the way that you do clinical trial design for these therapeutics. So from that, you get natural history data, you can get outcome measures, and then that all leads to human subjects research and clinical research. So why is basic research important? This is a quote from a French abbot of the 12th century. I thought it was funny. He says, "Some seek knowledge for the sake of knowledge, and that's curiosity. Some seek knowledge that they may be known to have knowledge, that is vanity. And some seek knowledge that they may give to others their knowledge, and that is charity." And basic science research is important because we can build our foundation of knowledge, but also we can apply that basic science knowledge to help diagnose, prevent, and treat disease. And that's really the core mission of what academic medicine is, meaning medicine as it's practiced at university settings. So Seeking knowledge for the sake of knowledge is what research is about. Seeking knowledge to give others knowledge is what education is about. And then applying that knowledge to diagnose, prevent, and treat disease is really core to our mission of patient care and service. So what are the big questions in basic epilepsy research? And I guarantee you I won't answer these questions for you today, but these are the things that a lot of basic science researchers focus on. So the first is, what causes epilepsy? It's the why question, the question that we get asked in clinic all the time. Why does my child have epilepsy? What causes epilepsy? And just to take a step back, epilepsy, as we define it, typically means a brain that is more susceptible to having repeated seizures than the typical population. So everyone's brain can have seizures if the right conditions are present, but some people's brains are more likely to have seizures, and then we call patients in that condition having epilepsy. And there's this whole process that the brain goes from not having epilepsy to epilepsy, and that's the term epileptogenesis. So genesis means the start of. So how does that process of a brain transforming in a way or having its neural circuitry and networks change so that it goes from a state where it doesn't have seizures that are not provoked to a situation where they have unprovoked seizures? And that's actually a very different question than why do seizures start and stop, which is what we call ictogenesis. So ictus typically means an event, and genesis, again, is to start. So what starts these events of seizures, and what stops them? Which is kind of more of a question of what transitions a brain that either has or doesn't have epilepsy in and out of these states of having a seizure versus not having a seizure. Also a very important question in basic epilepsy research, because a lot of the focus on therapeutics is to prevent seizures, to predict seizures, or to stop them with rescue medications after they start. Third is, what is the effect of chronic seizures on the brain? What happens to the brain in the situation of having chronic seizures? Are there different cells that are being produced? Are there different cells that are being that degenerate? Are there different network changes that happen with chronic seizures? Fourth, what are diagnostic and prognostic biomarkers for epilepsy? So I guess take a step back. What is a biomarker? A biomarker is basically something that you can measure that is helpful in either predicting disease, predicting the response to medications, or giving you some sort of a prognostic indicator, meaning helping you predict the future and what's going to happen with the course of disease. For example, right now we honestly don't really have great biomarkers except for EEG, but that's probably our best one. If a child comes into clinic and they've had a single seizure of their life and they otherwise don't have any developmental challenges, if they have an EEG that's normal, then they have a relatively low chance of having additional seizures. But if they have an EEG, even in between their seizures, that has a lot of spikes and sharps, signs that the brain is irritable and more prone to generating seizures, then that's a biomarker that they might have, that they're likely to have additional seizures in the future. And it might be even enough to trigger the clinician and family and child to start on anti-seizure medications, even after a single seizure. So biomarkers are an important one. And we're looking into a lot of different blood markers or markers in the cerebral spinal fluid, or imaging markers and other things that can be helpful to help predict response to treatment or prognosis for epilepsy. And then finally, importantly, what treatment approaches can prevent seizures or cure epilepsy? So with the knowledge that we have, what approaches work best? Meaning, when should we be treating? With what treatment modalities should we use? And how can we prevent seizures that way? So other people have thought about these questions as well, and there's been publications, including this excellent one from a few years ago, about what are the big questions. And again, they thought that the cause is important, this process of epileptogenesis is important, the epilepsy itself, and looking at clinical imaging and cell models and all this data that we have from a lot of different patients, and all that together can hopefully lead to cures, whether that be precision pharmacology, meaning drugs that are kind of designed for specific diseases or even specific individuals, cellular-based therapies, gene therapies that are potentially virally delivered, and epilepsy surgery. So what do we study in my lab? We are really interested in how genetic variants result in altered brain development and epilepsy. So we know that there are a lot of different genetic diseases that are associated with epilepsy, and a lot of genes that, when changed, can cause epilepsy. And how does that actually result in epilepsy? We don't know those processes as well. So we look into what cell types and molecular pathways are altered in this process to cause disease, and how can we treat altered cells and pathways to restore normal function. So the goal is always at the end, after we understand this, then how can we use that information and leverage it to treat the epilepsy? And our research, I view it as a circle, and the circle is to look at mechanism of epilepsy to advance precision therapies for epilepsy. So we always start with our patients, and from our patients, we've learned that there are a lot of DNA or genetic changes that can cause epilepsy. So that's kind of the root cause of the epilepsy, which is great. And that's been knowledge that's been advanced in the last few decades. The big gap, though, is although we know the genetic change, we don't necessarily know how that genetic change leads to altered cellular and molecular function. But if we can figure that part out, then the hope is we get targeted therapies, which we can then give to the clinicians who can do clinical trials, take that back to the patients, and that can inform what is happening when the DNA change causes epilepsy. This is a figure taken from a recent paper just showing how many gene variants are now known to cause epilepsy, where each of these rectangles is a single gene. SCN1A is highlighted here because it's one of the most studied genes in epilepsy. This is the gene that encodes a voltage-gated sodium channel. That's a component of the cell that lets in sodium in response to a cell reaching a certain voltage when it fires, like a neuron having firing. And that mutations in this gene can cause Dravet Syndrome. But there's a whole host of other genes, and there's continuously additional genes being discovered to be associated with epilepsy. And this is kind of a map of how many publications or how many studies are done on each of these genes. And it kind of gives you a sense of what the overall landscape of the research in genetic epilepsies is right now. So for example, the sodium channels are all here, potassium channels are here. So this whole block here is ion channels. These are different molecular pathways. I will be talking about the mTOR signaling pathway today, which basically encompasses this area here. But there's a bunch of other different cell types, or sorry, molecular pathways involved. So there's catalytic activity or enzymes. There's regulation of the formation of RNA from DNA, which is called transcription. There's transport of molecules across the cell membrane. There's things that are involved in cells differentiating in the brain. That means going from stem cell to neurons, for example, and a whole host of other categories. So my research focuses on the mTOR pathway. And why is that important? So we know that gene variants that hyperactivate this pathway called mTOR causes brain malformations and epilepsy. mTOR is the central regulator of metabolism. And what feeds into this regulator is if there's insulin, which reflects a fed state, nutrients, growth factors, energy, and amino acids like proteins in your system, in your cells, then mTOR is activated, and that leads to cell growth and protein synthesis. If your cells are starved for energy or have no growth factors, no nutrients, no amino acids, then this shuts down to basically protect the cell. Says in that case, it doesn't want to be growing or having protein synthesis. But what we now know is that there are many genes in this pathway, including STRAT alpha, the one that we study, that when they are changed, then mTOR activation goes up and no longer actually responds to these cues, and it's just hyperactive all the time. And that leads to brain malformations, including an enlarged brain called megalencephaly, enlarged half of the brain called hemimegalencephaly, or an enlarged focus in the brain called, a malformed focus in the brain called focal cortical dysplasia. And each of these is related to epilepsy, which can often be refractory to treatments with medications. So what happens in the disease we study, which is called PMSE syndrome, or polyhydramnios megalencephaly and symptomatic epilepsy. It's a mouthful, but these children that are affected have large heads, quite significant developmental challenges, and severe epilepsy. When you have a loss of this gene, then because of misregulation, then mTOR goes up and you get megalencephaly and epilepsy. So we use or I should take a step back and say that there are lots of ways to study genetic disease in epilepsy. A lot of different models that you can use. So one, you can study the human tissue directly, but that's often not feasible for various reasons. You can introduce those DNA changes into different organisms, including fruit flies, rats, mice, or fish, and there are epilepsy models for those. The approach that we take, because we want to actually look at human brain development and how that changes things, is we use human pluripotent stem cells. So that involves taking cells from humans, so either skin or blood typically. This is a picture of skin cells, fibroblasts from the skin. And then we add 4 genes to that, which reprogram these cells into stem cells. And then we get a cell type called induced pluripotent stem cells, which theoretically are able to generate any potential cell in the human body. They're nice to use because they're patient-specific, meaning if the subject that we take the cells from have a genetic change, then their stem cells will also have that same genetic change. We can make them generally pretty quickly, within 3 weeks or so. And we can also use gene editing techniques to fix the mutation in the cell or introduce a mutation in an otherwise normal cell to study the gene changes. And these induced pluripotent stem cells, again, can be made into any cell type, and they also self-renew, meaning you can grow them. They reproduce in culture, so we have a basically never-ending starting material to study different pathways. So from these stem cells, we are able to, in a dish, make them into neurons in a flat culture or 2-dimensional culture. We can make balls of cells, which I'll talk about more, called organoids, which recapitulate aspects of neural development. Or we can even study non-neural things like heart cells, cardiomyocytes, because some of the gene mutations affect the heart as well as the brain. And then once we generate these neural tissues, we can do functional and molecular studies. For example, we can record from the cells and look at their electrical activity using various ways. And then we can look at different molecular levels of proteins or RNA. That's, again, the substance that's made from our DNA, to see what changes are present in the patient-derived cells. So I mentioned these 3D organoids, and iPSCs can form cell clumps that resemble the developing human brain. And this is one of the early papers that took advantage of this technology. So what you get is neural tissue. So astrocytes, neurons, and they grow in 3D. So they're not the same size as a brain. They're probably more like 3 to 4 millimeters in diameter, but it's advantageous because the organization recapitulates some of the brain structures in the developing human brain. And just to remember, the human brain development is a lot different than a mouse brain. And the mouse brain development has been thoroughly studied, but is a lot harder to study the developing human brain. And there's a lot of differences between the human brain development and mouse brain development, not just size, but cell type, diversity, and just the shape and size of some of the cells that we have that rodents don't. So these organoids really give us a way that previously wasn't present before to watch a developing human brain-like structure grow in a dish. So when we grow these in the lab, we've typically used these spinning bioreactors. So just for scale, this is about the size of my hand, and there are 12 little propellers, basically driven by a motor, that help get the oxygen and nutrients circulating into the brain organoids. And what we did is we grew stem cells from individuals with PMSE and compared them with controls. And remember, these individuals with PMSE have larger brains. And we find that these PMSE organoids had a delay in forming neurons, meaning they stayed in the stem cell state for longer, which can explain how they have bigger brains, because their stem cells are self-renewing and they're replicating. And the PMSE organoids also had a much different shape here in PMSE compared to controls. And when you take a cross-section through, this is what they look like compared to controls. So a very striking phenotype that was present in the brain organoid system. We also found that later on at week 12 of development, they had an overabundance of a human-specific neural progenitor cell type. So I'm going to geek out for a minute about this specific cell type called outer radial glia. These ORGs are present in human brain development and in primate brain development, but not really present in rodent brain development. And their discovery was really interesting because one of the big questions in science is, why do humans have the complex, large brain structure that a lot of other animals don't? And one of the reasons we think is because we have this particular cell type that is a progenitor cell type that other species don't really have as much of. And this is what leads to the expansion of the human cortex versus other animals. And what's known is that in this particular cell type, the mTOR pathway, the pathway that we study, is highly upregulated in these cells. And now we found that when we don't have the STRAT alpha gene, which leads to mTOR pathway hyperactivation, we have an overabundance of this cell type. So this is another way why we think that the individuals with PMSE have an expanded brain and large brain compared to other typical human individuals. And finally, we also saw this particular cell type, which is known to happen in human brains with mTOR pathway hyperactivity. But for some reason, in animal models, it really has not been seen. But now we see these in our human stem cell models. So these are called balloon cells, which is a distinct cell type, which is very enlarged and often has multiple nuclei in them. And what we think is that because our model system is human specific, then we are able to generate this type of cell. Maybe even it's coming from these outer radial glia. And that's why the animal models for mTOR hyperactivity have not seen this important cell type. From our organoids, we can also look at neuronal function. So we can use different methods like adding a dye that changes in intensity with calcium concentration. So when neurons fire, they have an influx of calcium into the cell, and we can actually see on a single cell basis and over time networks of activity happening all together. So that's the video that's sped up about 6-fold, but this is a 2-minute recording over time. And if you take a few regions of interest and you plot them out, you can see that some of these cells fire all together, and then some things kind of fire separately. We also look at these microelectrode array recordings, which I think of as basically a tiny EEG on the surface of a organoid. And what you'll see is firing in 2D space, where bright red or white is a lot of firing, and then blue is less, and black is nothing. And you can also look at what we call raster plot, which is each electrode is on the y-axis, and each time there's a firing event, it draws a little bit of a dot. So when we have done this, we've seen that some of these PMSE organoids may have seizure-like events, and I don't want to oversell this because we haven't looked carefully enough at controls yet. But sometimes what we see is a big burst of activity, and then that kind of repeats in a periodic way, and then slowly tapers off, which is exactly what we see seizures look like on an EEG in patients that we've monitored in the EEG lab. So we hope that these functional measures will give us an ability to screen different drugs to see what might be better at treating seizures and epilepsy. Our future studies in our lab look at inhibitory interneurons. So just as a step back, the brain mostly from the neuron side contains excitatory and inhibitory interneurons. So excitatory neurons are those that promote more activity. Inhibitory interneurons kind of regulate and decrease activity, and you need a balance so that you don't always have seizures. And one thought is that the inhibitory interneurons don't develop well with mTOR hyperactivity present. So we are looking at interneuron migration. So you can see here, this is a sped up video, but you can see live that there are cells migrating and jumping around. And then sorry, it's really jumpy, but there are outgrowths from the cells too that we can see as these interneurons find their way and develop. And we also can make fused organoids, meaning an excitatory organoid fused with an inhibitory organoid, and look at the migration of the red cells over to the green side. And we can actually fix that and do a three-dimensional image and look in 3D space the migration of the interneurons over from the ventral to the from the inhibitory over to the excitatory side. So that's one of our major focuses in the lab moving forward. So what's the impact of this research for epilepsy? Why should we care? Why does a taxpayer pay money to the NIH, which then we receive to do research for this? So we want to modulate aspects of the mTOR pathway and related pathways to prevent seizures and progression for epilepsy. Just remember that right now there really isn't, except for everolimus, which is an mTOR pathway inhibitor, there really aren't medications that get at the underlying process of epileptogenesis, meaning the medications present that we have in our armamentarium approved by the FDA really suppress seizures, but they don't prevent the progression of epilepsy or prevent epilepsy from happening. If you take those medications away and you still have epilepsy, then your seizures will recur. What we hope to do is really get at underlying disease-causing mechanisms to prevent the progression of epilepsy. And everolimus has been a great step, but it's not been perfect, meaning it's not really been able to suppress seizures much more than any of the other medications. What we think is happening is that there are a lot of related pathways that then kind of get upregulated when patients are on everolimus. So we're exploring those aspects. We also just are really interested in how does the brain form. And using this model, we can finally have a way to really look at early events of brain development, human brain development in a dish, and manipulate that and study that over time. And then finally, we would love to have a human cell model to study the mechanisms of epilepsy and test treatments for seizure in a dish, because those are much more manipulatable than mouse or other animal models potentially. So this is my team. Research is best done in a team. Grace is a PhD scientist. Shivanshi has left the lab, but did a lot of this amazing work that I showed you today. And then we have another advanced technician, Daniel Shaklik, who's headed to grad school, and then Sahej is our undergraduate student. And I should also note that the University of Michigan has a huge group of basic science epilepsy researchers. And when you look across the country and the world, it takes a large group of people with a diverse set of interests to really study epilepsy, from the molecular side to the genetic side to the animal model, stem cell model, computational side. And this is just part of the major group of researchers at University of Michigan. Laurie Isom and Jack Parent are two of the top researchers in the country for epilepsy. And Jack Parent's my research mentor. Laurie Isom is also a strong collaborator of mine. And then in pediatric neurology, Julie Zebro is also working in genetic epilepsies, and Karin Smith is working on a computational way to look and see whether there are biomarkers on intracranial recordings that can give rise to accurate epilepsy surgery resections that are successful. So I'm going to tell you about two stories of research done at U of M that's been really, that's either been transformational or has the potential of doing that. So the first is on Dravet Syndrome, which is, again, one of the most studied genetic epilepsies. And what's known is that we each have 2 copies of every gene. So we typically have 2 copies of SCN1A. And this gene, again, encodes this protein that lets in sodium ions into the cell, that helps them fire. And in individuals with Dravet syndrome, they have a loss of one copy of SCN1A, or at least dysfunction of one copy of SCN1A, and that leads to fewer functional sodium channel units, which leads to seizures and cognitive and motor impairment. And what a company has found previously is that you can use these compounds called antisense oligonucleotides, which are basically short components of DNA, RNA type molecules. And with this very fascinating approach, they actually bind to an element within this gene, which can then lead to an increase in production of SCN1A molecules. So their big question is, okay, well, we found this element in SCN1A, which we think we can modulate the splicing of this gene, and that can lead to potentially more sodium channel units. And would that actually work? So they approached people at University of Michigan, including Dr. Isom, who has a mouse model for Dravet syndrome, and they started doing injections into the spinal fluid in the brain of this compound at 2 days after birth. And typically these mice, these Dravet syndrome mice, have a high mortality around 3 weeks of age. You can see when they're treated with a vehicle control, there's a lot of mortality. So going down means that there's less survival over time. But the mice that were injected with this compound, whether they had a genetic mutation in them or they were without the genetic mutation, had a significant prolongation of survival. So it was a pretty amazing result that was published 2 years ago. And with that data brought to the Federal Food and Drug Administration, it's made it into clinical trials. So even that same year, the first human patient was injected with that antisense oligonucleotide. And our research group, led by the principal investigator, Dr. Joshi, and Julie Zebro as the co-investigator, and Stephanie Rao is our clinical research project manager leading this leading the trials at University of Michigan. So we're one of the sites in this national or international trial for an antisense oligonucleotide. Another very interesting genetic epilepsy story is one of the other frequent causes of genetic epilepsies in females is caused by variants in a gene called PCDH19. This is a fascinating mechanism because it's a really unique cellular mechanism. So typically when we think of a gene variant and you have loss of one copy and you have disease, you would think that loss of two copies is even worse. It's not the case with PCDH19. So this happens to be an X-linked gene. So just to review, males are XY. We have an X chromosome and a Y chromosome. Females have two X chromosomes in the typical condition. This PCDH19 gene is on the X chromosome. And interestingly, females that have one affected chromosome, one mutant chromosome, and one typical chromosome are affected by the disease. They have these clustering seizures and developmental challenges. But males who only would have PCDH19 on the X chromosome, and if that carries a mutation and we don't have a copy of PCDH19 on the Y chromosome, are actually unaffected. So it's this weird thing where it's not a dose dependent thing, but it's really when you have the presence of a normal copy and an abnormal copy, they seem to interfere with each other, and that's what causes disease. What Dr. Zebro has shown in her mice and Dr. New has shown in an organoid model is that these cells, whether they have the gene or don't have the gene, segregate out during brain development, causing this really striking striped pattern in the developing brain, where those that have the mutation cluster together and those that don't have the mutation cluster together, likely causing changes in network development and likely resulting in the seizures in the animal models as well as in humans. We have a lot of clinical research going on in our pediatric neurology group too, and these are the principal investigators involved in that. We have a robust clinical research coordinator team that does all the hard work for that research. I mean, we have a lot of questions that we're trying to answer, including natural history studies and clinical trials for Dravet syndrome, like I talked about. For example, how do sleep and epilepsy affect each other? National databases for epilepsy genetics and epilepsy surgery. How do we improve equal access to epilepsy surgery? How can quantitative EEG help us improve care in children? So this is a machine processed EEG, time compressed EEG that can, using computer technology, help us figure things out on EEG that the human brain and eye can't quite see. How do we leverage a learning health system for epilepsy? So the idea for this is all of our patient data gets fed from multiple centers into a central database. It gets processed, and then all those health systems can learn from each other and from each other's patients about what the optimal treatments or prognosis is for epilepsy patients. What biomarkers predict response and relapse for infantile spasms, and what are the best treatments for children with Lennox-Gastaut syndrome? We have also robust neonatal seizure research, and that was spearheaded by Renee Schalhaas. So for neonates who have seizures, what are early childhood outcomes? What are genetic factors? And which neonates go out to have chronic seizures or epilepsy? And what inflammatory markers might predict risk of post-neonatal epilepsy? There's a lot of research outside of U of M, of course, too. So some of the things that I've been most fascinated by and have been trying to follow pretty closely is interneuron transplantation for epilepsy treatment. So remember, the interneurons are the inhibitory cells that can kind of dampen the neuronal activity and control seizures naturally. And we know that there's loss of these interneurons in a lot of epilepsy, so maybe we can transplant them back into the brain to help seizures. If you ever are curious, to follow on Twitter, Cronut1, this is a sea lion in the San Francisco Bay Area that was poisoned by domoic acid, which is a naturally occurring neurotoxin that forms from red algae, if I'm not mistaken. And what Scott Baraban, a researcher at UCSF, did in collaboration with some neurosurgeons there is they put Cronut on the operating table at UCSF and injected porcine-derived, meaning pork or pig-derived interneuron cells into Cronut's brain, and since then Cronut has been seizure-free. So this is kind of one of the first large animal proof of concept studies to show that that's a potential treatment that might be able to be translated to humans sometime, hopefully in the near future. There's a group at University College London that are using closed loop gene therapy to quiet down overactive neurons. So their idea is that, you can make neurons quieter by introducing genes that quiet down the electrical activity. But the problem is you don't want to quiet down all your neurons because you also need your neurons to fire to think. But their technology actually only takes the overactive neurons to drive that gene to be expressed to quiet it down. So it's kind of a responsive system, a closed loop system where the only neurons that are being quieted down are the overactive ones, hopefully controlling seizures. And they've shown this in rodent models that it can control seizures. And Dr. Heather Mefford at St. Jude Children's in Memphis has done amazing work in the past and continues to do amazing work on defining the genetic underpinnings of epilepsies, and really has, she and a lot of other colleagues around the world have really defined the field as really showing, okay, here are some of the key things that can cause epilepsy, meaning the key genetic underpinnings to what causes brains to potentially have chronic seizures. So how long does it take to translate basic research into clinical therapies? It typically takes 12 or more years, but it can be faster. So we think of this as this pyramid of drug discovery, which might have 1,000+ compounds, and then finally get to one compound with FDA approval. And there's a lot of drop off with each step here. Costs billions of dollars or more, and it can take 12 or more years. That said, things can happen more quickly sometimes. So I like the example of Dravet syndrome, where it was described clinically in 1978. The gene was found in 2001. The mouse model was in 2006 published. Stem cell model published by Jack Parent's group at Michigan in 2013. And then in the last 5 years, there have been specific drugs that get FDA approval for Dravet syndrome. And from the mouse study done by Laurie Isom's group here, published in 2020 for the antisense oligonucleotide therapy, the clinical trials started shortly thereafter, approved by the FDA. Or not, the drug's not approved by the FDA, but the trials were given the go-ahead by the FDA. So what is the impact of this research for epilepsy? Oh, sorry, that's a redundant slide. I think that's the end, actually. Thank you.
Okay, thank you so much, Dr. Dang. That was absolutely fascinating, and really gives a lot of hope to the whole epilepsy community, just all of these exciting discoveries on the horizon. So we have a good 10 minutes or so for questions. If anyone has any questions, you can either unmute yourself and ask a question, or you can type a question into the chat. So I'll give some people some time to think. If no one has questions, I have a few that I can ask. So
yeah, that's a great question. So so I guess to take a step back, spikes on an EEG I think of as like a spark, whereas a seizure on an EEG and clinically I think of as like a fire, to make a bit of analogy, meaning a little bit of a spark may not cause any problems in itself, but it might be an indication that there's some irritability in that brain and that it might be more prone to having a seizure. I think the other way I kind of think of it as, if you think of a stadium of people and when they do the wave in the stadium,
Yeah.
sometimes it's just like a single person that puts their arms up and then it doesn't really go anywhere. Sometimes a group of people put their arms up, doesn't go anywhere. And then eventually sometimes a group of people puts their arms up and it starts this whole wave across the whole stadium. So I think of it as when it's just a single person doesn't go anywhere, or just even a group and doesn't go anywhere, that's like a spike or a sharp in the brain. But it doesn't lead to this prolonged thing that is a continuous thing, not continuous, but like at least for some period of time it continues, and that wave going around the stadium is more like a seizure. So it's possible to have spikes on the EEG without epilepsy, but it certainly, if you have a lot of spikes, it certainly pushes us to thinking, okay, well, this person is very prone to having seizures, and they might be having seizures that we're not quite recognizing all the time when you're just looking at someone, because seizures can look like all sorts of different things. And it doesn't always have to be a grand mal type seizure where it's a generalized tonic-clonic seizure where there's bilateral shaking and stiffening of the arms and legs. There is a basic science question, of course, of whether those spikes cause an issue in themselves. And generally, it's thought that if you have a lot of spikes, then there is evidence that you can have very, very brief altered awareness and delayed response time when that spike happens. And there's also some evidence that people with a lot of spikes may have some delay in learning compared to someone without spikes. That said, it's very tricky to think about what that means clinically, meaning, do we then try to treat a patient so that we get rid of all the spikes? Or do we just try to treat them so that we get rid of the seizures, but it's okay if they have some spikes? And in most cases, we are okay with just treating for seizures because there's a balance. If we try to get rid of all the spikes, that usually means we usually, one, aren't able to, and two, might mean going up on a bunch of different medications, which might also lead to side effects. So there's a line that we have to draw, and we don't want to overtreat patients either. And there's one specific circumstance where sometimes we treat the spikes themselves, and that's in a condition called ESES, or electrographic status epilepticus of sleep, where in some individuals, they don't have a ton of spikes when they're awake during the daytime, but then when they fall asleep, those spikes are nearly continuous. And what we found is that in some of those individuals, they have problems with learning or behavior or even regression, meaning they used to be able to do things and now they can't anymore with those spikes coming about. And that just means that their sleep is really disrupted because they're having the spikes all the time. In those cases, we might actually treat the spikes themselves.
Okay, well, while people are thinking, I have a question. You talked a lot about genetic epilepsies. What type of basic research is being done for epilepsies that are caused by some sort of trauma, such as a traumatic brain injury or stroke? I imagine there may be research looking into inflammation and kind of the after effects of those injuries and how that leads to epileptogenesis. Can you talk a little bit about that?
Yeah, definitely. Thanks, Russ, for that question. I realized that my talk was very genetics heavy, but we, of course, have a lot of people with epilepsy that have acquired causes of epilepsy, meaning, like you said, from hypoxic ischemic damage, meaning lack of oxygen or blood flow to the brain, whether that's in the neonatal period or from a stroke in the neonatal period or afterwards, or traumatic brain injury. A big area of research is in the traumatic brain injury, because that's another model where you have epileptogenesis, meaning you can have seizures right when you get your brain injury, but often the thing is, what often happens is you might have a seizure or two with the brain injury, but then you have this period where you don't have seizures, and then later on you have epilepsy come out with chronic seizures. So there's that period of epileptogenesis after traumatic brain injury, and there's a lot of active research trying to figure out, one, can we determine at the outset which patients will go on to have epilepsy after a traumatic brain injury versus those that don't? And can we find biomarkers of epileptogenesis during that process? Because if we do, then maybe there's something that we can do with regard to controlling inflammation or other proposed mechanisms for epileptogenesis during that period. Can we have interventions during that period for the high-risk patients? We wouldn't necessarily want to give all the patients with traumatic brain injury treatment to try to prevent epilepsy from occurring, but for those that have biomarkers that show us that they're at high risk, perhaps that's the group that we can intervene with. And same thing with neonatal seizures and hypoxic ischemic damage. A lot of the research is trying to look at neuroprotective things that can be done in that period. One of the big things that have come out, which we do routinely now, is with therapeutic hypothermia, or just basically whole body cooling of the neonate who has suffered from hypoxic ischemic damage during or before the birthing process. So that's a routine thing that we can do now to help neuroprotect and hopefully improve seizure outcome later on too.
And there's a question about non-epileptic seizures and research into that, and I'm not sure if that's referring to psychogenic non-epileptic seizures, but that certainly is an interesting area of research as well. Do you have any idea whether there's is research in that area?
There's definitely a lot of research being done in that area too. So there are different terms for this: non-epileptic events, functional neurological disorders with seizure-like episodes. Some people just call them non-epileptic seizures. And what we've found, or what not we, but people have found now, is that there are biomarkers for that as well, meaning there are actually structural brain changes in individuals with non-epileptic events compared to typical controls. So there's definitely there's a biological basis for these non-epileptic events too, which I think has really energized the community of people that suffer from these disorders, because they've sometimes, again, not correctly, but they've sometimes been told this is all in your head, it's something that you just need to get over, blah blah blah. But I think it's validating in that it's showed that there's actually stuff going on biologically that is related to non-epileptic events.
Great. Well, again, thank you so much for an excellent presentation. And anyone, this will be added to our website, and to the CS Mott Children's Hospital YouTube channel, so people can watch this afterwards and go back and remember some of the very interesting things that you talked about. And so keep up the great research, and thanks so much, everyone, for participating today.
Thank you, everybody, for coming. Thanks, Russ. Appreciate it.